Namaste to all students. Kusho Bitya, myself Jayoti and Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about plant regeneration. And this session related to plant biotechnology course of program MSc BT second sem. The process of growing an entire plant from a single cell or group of cell is known as plant regeneration. Regeneration is possible because plant cells can be made totipotent using hormones. Differentiated tissues like stem, leaves, root, etc. and undifferentiated means embryonic cells are totipotent. And it can become a whole new plant by differentiating into a new whole plant. The plants can be regenerated by organogenesis process or by somatic embryogenesis. If we talk about the organogenesis, so it refers to the formation of organs from the cultured plant. The shoot buds or monopolar structures are formed by manipulating the ratio of cytokinin to auxin in the culture. In second type, which is somatic embryogenesis, so in somatic embryogenesis, the totipotent cells may undergo embryonic pathway to form somatic embryos, which can grow to regenerate into complete plant. If we talk about the plant material, the section of plant, it can be shoot, it can be leaf section, root, or reproductive organ of flower. So these segments of plants which are going to use in plant tissue culture are known as explant. So any part of the plant taken out and grown in test tube under sterile conditions in special nutrient media is called explant. Here you can understand about the concept of plant tissue culture by this simple flow chart. This is plant, so you can collect the explant, then you can inoculate this explant on nutrient media, which will create further callus and it go for the somatic embryogenesis. So it will first create embryoid and then embryo, which further go for regeneration process. Means you have to shift media with the shoot culture media and root culture media, by which complete plantlet will be grown. Now this plantlet will have to shift in field. Hormones which are used in plant tissue culture media, so auxin, cytokinin, gibberellin, abscisic acid, and polyamines can be used. And if we talk about the environmental conditions, so you have to use nutrient medium like inorganic salts, a carbon source, vitamin, amino acids, and growth regulators need to be used. And aseptic conditions have to maintain, so the microbes compete with growing tissue and finally kill it. It is essential to maintain aseptic conditions of tissue culture. Thus, sterilization means complete destruction or killing of microorganisms so that complete aseptic conditions are created for in vitro culturing. Third step, which is aeration of the tissue. So proper aeration of the culture tissue is also an important aspect of culture technique. So it is achieved by occasionally stirring the medium by stirring or by automatic shaker. In regeneration of plantlets, there are various steps which has to be followed. First one is preparation of suitable nutrient media, then selection of explant, then sterilization of explant, and then inoculation and incubation, regeneration, further hardening, and plant-led transfer. So in preparation of suitable nutrient media, so suitable nutrient medium as per objective of the culture is prepared and transferred into suitable containers. Then selection of the explant. So selection of explants such as shoot tip should be done. Further, sterilization of explant need to be done, and it can be done by disinfectant, and then washing of the explant with the sterile distilled water is essential. Fourth step is inoculation. So inoculation means transfer of the explant into the suitable nutrient medium, which is sterilized by filter sterilized to avoid microbial contamination in culture vessels under sterile condition is done. Fifth step is incubation. So growing the culture in growth chamber or plant tissue culture room, having the appropriate physical condition, for example, artificial light, 16 hours of photo period, temperature should be in range of 26-degree temperature and relative humidity of 50 to 60% is required. Sixth step is regeneration. So regeneration of plants from cultured tissue, plant tissue is carried out. And seventh step is hardening. Hardening is gradual exposure of plantlets to an environmental conditions. Eighth step is plantlet transfer. After hardening, plantlets transfer to the greenhouse or field conditions following acclimatization, means hardening of regenerated plants. You can understand regeneration process of whole plant using tissue culture technique by this flow chart. So here I'm sharing the screen, and in which first of all you have to select plant, mother plant, by which you will collect your explant. Then sterilization and growth of on suitable media of explant need to be done. Then multiplication in test tubes will be done of plantlets, and further it has to go for hardening, which is followed by transfer of the to the soil. And that's how you can collect number of plantlets by a single explant by using tissue culture technique. Next step is plant propagation. So propagation of plant is defined as production of new individuals from a selected plant having all the characters of the original one. New plants are created from a variety of sources like seeds, cutting, bulbs, tissue culture, etc. Plant propagation is done sexually or asexually as well as artificial platform using tissue culture techniques. What is the principle behind the plant propagation? So the basis of plant propagation is totipotency. Totipotency is the capability of cells to regenerate missing parts and subsequently an entire organism. In plant propagation, it can be done by sexual, by seeds, and or asexual or vegetative by cutting, grafting, layers or suckers, and micropropagation. So in sexual propagation, it is with the use of seeds or spores that is separated from the parent plant. This method is so termed sexual because there is the involvement of the sexes referring to the contribution of both male and female gametes in the production of new plants. Pollen is transferred from anther to stigma and fertilization occurs and seeds are produced. Here you can understand the natural process of propagation by this flow chart. Here is a flower which is having both male organogenesis organs and female organs. This is stamen and this is ovary which is having ovules, and stamens is having anther which is filled up with the pollen grains, and pollen grains actually containing the sperm nuclei. During the pollination process, pollen will stick to the stigma and they will be germinated and further fused with the egg, and they release their male gamete to the egg, and which create the embryo, and fertilization eggs become the embryo to of the seed. This is seed you will get after the sexual propagation, and these seeds can germinate to produce a complete whole plant. In asexual propagation, it is also called vegetative propagation, is with the use of planting material which are vegetative parts of any plant rather than seeds or spores which are reproductive parts. For example, cutting, layering and grafting. Here you can understand it by this diagrammatic example. You can cut this region of plant by blade, and this is the cut at the slant. You have to create actually slant, and then you can grow this cutting region into sand or soil of compost. Tissue culture also be a part of asexual propagation. It is also called in vitro culture and micropropagation. It is a technique of propagating plants indoor under aseptic or sterile conditions in artificial growth medium. The growth medium is also called nutrient solution. It is a mixture of essential elements like macro and micronutrients, vitamins, hormones, and carbon source. And the initial propagule in tissue culture is called explant, by which you can create complete whole plant. So, micropropagation is the propagation of plants through tissue culture. It is a proven mean of producing millions of identical plants under controlled and aseptic conditions, independent of seasonal constants. Small pieces of plant tissues means explants used for regeneration and shoot system rooting and growing into full-size plant. So starting material we need to use is tip bud, leaf, axillary bud, internodes, roots and seeds. And if we talk about the steps of micropropagation, so you have to follow these 5 steps like selection and preparation of mother plant, then initiation of culture, and third one is multiplication, fourth is rooting, and fifth is transfer to soil. So first of all, you have to go for the establishment, which is the objective of this stage is to get clean cultures that can begin the process of shoot multiplication. And the explant is usually sterilized with a combination of detergent and bleach. In difficult situations, alcohol or a fungicide may be used. Second step is initiation. So explant placed in a growth medium. And third stage, which is multiplication. So explant transferred to the shoot media where shoots can be constantly divided. The objective of shoot multiplication stage is to increase the number of shoots produced by original explant. The culture or a portion of the culture is removed from the jar and placed on a sterile paper towel. A scalpel and forceps are used to cut and separate the larger culture into smaller pieces for transfer to a new jar to complete the subculturing procedure. A high cytokinin to auxin ratio is used during the multiplication stage to induce axillary and or adventitious shoot formation. This ratio is decided upon the preliminary research, and too high a concentration of cytokinin will result in a high number of adventitious shoots that do not elongate. And common cytokinins used in culture are benzyl adenine, BA, and kinetin. Fourth step, which is rooting. So shoots multiplied in culture must be rooted in order to create a new plantlet. In the rooting stage, micro cuttings are induced to form roots, usually by application of auxin hormone. Micro cutting can either be rooted in vitro or ex vitro. In general, microcutting rooted ex vitro have a more normal root system and acclimatize to a normal growth environment better than cutting root in vitro. However, the propagator has more control over the rooting environment in vitro, and this method may fit their production scheme better. You can differentiate in vitro rooting and ex vitro rooting by this simple photo. And micro cuttings are inserted directly into the rooting substrate, often using forceps to handle the small cutting. Fifth step, which is transfer to the soil. So finally, after roots have become well established on the micro cutting, plantlets must be acclimatized to a normal growth environment. This involves gradually moving to open air conditions where the humidity is reduced and light level increased. What are the applications? So it is used for the rapid multiplication, seasonal independent production, used to conserve rare or endangered species, and greater output. Next topic or next session is somaclonal variation. So the term somaclonal variation was first used by the Larkin and Scowcroft in 1981. Somaclonal variation is the variation seen in plants that have been produced by plant tissue culture. The term somaclonal variation is used for genetic variability present among all kinds of cells or obtained from cells cultured in vitro. Plants derived from such cells or progeny of such plants is called somaclones. These variations can be detected as genetic or phenotypic traits. What are the basic features regarding somaclonal variation? So, variations in number and structure of chromosomes are commonly observed. Regenerated plants with altered chromosomal changes often show changes in leaf shape and color, growth rate and habit, and sexual fertility. It is generally heritable mutations and persist in plant population even after plantation into the field. Here I am sharing the screen to explain the somaclonal variation by this flow chart. So this is callus tissue which can go further for organogenesis. By the organogenesis, you can generate the plants and after generating plants, you can go for the hardening process. In hardening process, you can simply see some variation from mother plants. So you can select those somaclonal variants and then further you can go for again in vitro culturing of these somaclonal variants. What are the causes of somaclonal variations? So the causes may be physiological, genetic, and biochemical. If we talk about the genetic reason, so pre-existing variations in the somatic cells of X plant that are caused by the mutations and other DNA changes. Typical genetic alterations are changes in chromosome number, which is also known as polyploidy or aneuploidy. Second is changes in chromosome structure, which can be done by the translocation, deletion, insertions, and duplications of the gene. Third one is the DNA sequence. So mutation can done the DNA sequence alteration and it occurs at the high frequency. Second reason, which is physiological, so exposure of the culture to the plant growth regulators for long term can create the somaclonal variants, and culture conditions also affect the somaclonal variation formation. Third step is biochemical. So lack of photosynthetic ability due to alteration in carbon metabolism can cause the somaclonal variants, and antibiotic resistance also can create somaclonal variants. What are the advantages of somaclonal variants? So rather stable and high frequencies, new alleles or even novel mutations may be isolated which were not available in germplasm. It can be performed in all type of plants and it reduces the time required for the release of new variety by 2 years as compared to mutation breeding. It is a very effective selection method. Along with the advantage, there are some disadvantages also regarding somaclonal variation method. So it is applicable only for those species cell cultures which regenerate into complete plants. Selected cell lines often show reduced or no regeneration potential. It shows undesirable features such as reduced fertility, growth rate, and even overall performance of the plant. Selected clones of somaclonal variants are unstable, not always stable, and heritable. So these are some disadvantages regarding somaclonal variation. So now this is the sum up of this session, and this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.